So there's a huge need to feed good data into those models. And I don't know, but obviously there's a lot of hype about AI and machine learning, etc. But we'll see what will come out of this in the next 5 years. I'm really curious to see what will be possible. I don't know a lot about it, but we'll try to help with our assays to really make those tools more powerful, and we'll see how that develops.
Hey, smart biotech scientists, welcome back. We are in the middle of a conversation on high-throughput workflows to screen critical quality attributes in the developability assessment and process development. And my guest today is Sebastian Giehring, who is the CEO and founder of PAIA Biotech, an innovative leader in bead-based assays. If you haven't listened to part 1 yet, please go back. And today, we are continuing our journey. Stay tuned. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Can you help us understand, Sebastian, about what workflow is involved? Because yes, we have a technology that works great. It's a technology that allows to do high throughput screening. But what does this mean in terms of sample prep or the use of the plate or the reader, and I'd say in a more global picture? Can you walk us through a standard workflow for these assays?
In the best case, I guess would be too long to address the different assays one by one, but in the optimal scenario, you would only need to take your cell culture sample, dilute it in like a normal standard buffer, adjust it in terms of concentration, and then run it directly in the assay. So one of my favorite examples, if we come back, for example, to the glycosylation screening, is a customer who runs a silation screenings and cell line development, and also in upstream development, for example, in amber bioreactors, and where the aim is really, with this simple approach, just in diluted supernatants, to achieve the degree of silation that they need if they have to match a certain reference product that they had from a previous process. This is the ideal scenario, right? Just to be able to do this from diluted supernatant. And that's what we're aiming for also with the aggregation assay and this purity mispairing assay.
Using the technology and knowing how to integrate is one thing, but we also need to talk about designing the whole experiment. So what are the do's and don'ts when designing a screening experimental plan? Because we have to coordinate the analytical part. You mentioned cell culture. There might be other factors we have to take in account. From your experience, what are the do's and don'ts?
This is a tough one. What we see usually is the analytical side. And what we would always emphasize is that you need to have well-defined reference material. So if you're starting a project for a certain type of molecule, you need to have some references. For example, you need to have data on your glycosylation, maybe to have different types of this product with different types of glycosylation, or you need an aggregation standard. If you're looking at aggregation, it's very tricky. Aggregation also can change over time, depending on how samples are stored, etc. So it's really about having well-characterized reference materials so that you know what you can expect. And if you don't have material available that you can run alongside our screening assays, then it's kind of, you don't know exactly where you are. And this is, of course, always tough to interpret results if this is not clear. And I think on the cell culture side, I guess you know more about it than me. I'm not a cell culture person, but I guess there are a lot of little things that you have to pay attention to make sure that these processes are producible and you try to control them as much as possible.
Definitely, that's my advice as a cell culture scientist and engineer. You need to design your experiment well so that your runs are robust, your runs will lead meaningful results, and also very importantly to induce enough variability in your experimental conditions so you can then use the data you're generating and leverage that to the most. Yeah, because what you want to do is not just looking at your data, you want to get some information out and ideally even create some models then you can use to make informed decisions for maybe a future experiment or a process validation or whatever it might be.
Yeah, I mean, that's what we also see in our assay developments here in-house, that you sometimes have to vary the parameters by more than you think we usually do, because then you get to find some effects that you may not have found. I mean, we're talking about DOE, for example. So you may find some effects that you don't find if you just run your cell culture in the way you've always been doing it. So if you have the resources to do this, I guess that's the way to do it. And of course, you have to master these methods.
Now, speaking of resources, that's a good point, Sebastian, because some of our listeners work in small companies and they may have a limited amount of devices at their disposal. What is the minimum requirement, let's say, for a reader of your assays? What kind of readers could they use?
There are two ways. One thing is a normal plate reader from one of those well-known brands that can do a fluorescence from the bottom. These are instruments which are not usually used in cell culture labs, but most companies do have that somewhere else. But you can also use microscopes, basically, that have the fluorescence option in there. Like, for example, the machines from Cytina, Advanced Instrument, but also companies like Synentech, which have cell imagers with fluorescence. So all those machines are very well suited for the purpose. And that's machines that people have because they have to image cells anyways. And if there's the fluorescence capability in those instruments, they can also use those machines for our assays. And besides that, they only need an orbital shaker. That's all.
That's fantastic. Sebastian, can you share some real-world examples where you saw how high-throughput workflows significantly improved the development workflow and also the outcome of these experiments?
We see most benefit of these screening technologies when it comes to projects where you really know the target profile. So be it in terms of glycosylation, so that these assays really can, for example, when we're talking about biosimilars, right? Then you know exactly what you're aiming for. And if then you have a couple of cell lines that you have to tweak by media optimization, for example, and you have to test a lot of different kind of supplements to try to tweak the glycan profile of the product, then our assays are really very suitable and they provide the throughput that you need to really try a lot of different combinations and finally find the cell line, the right cell culture conditions that allow you to have a really good quality product in the end. This biosimilars is a good example, I would say.
Let's speak about the developability platform for a while, because you have pioneered unique screening platform. Can you tell us on how this platform works and how it revolutionizes the whole screening and optimization?
This is something that we're doing a lot these days because this is a whole different game. It's still, we're using the same plate technology and the principle is the same, but we're looking at completely different parameters here. So the scenario is like people in discovery have a lot of different antibodies that they need to bring forward and they select for binding, etc., target binding, affinities and stuff. But at the same time, they need to look at other parameters which later on really impact the developability of a drug. So this can be biophysical parameters like stability, aggregation, propensity, etc., but also parameters that we address. For example, hydrophobicity is a big thing. Of course, it's also related to aggregation and stuff, but it's also about non-specific binding to non-targets. So that's called polyspecificity or polyreactivity. So we're talking here about interactions of the antibody candidates with some kind of proteins or cell surfaces, whatever you may find in the body that lead to binding or loss of the actual drug to something which is not the target. And it's very important to assess this very early on. And our assays are very well suited for this purpose because these are pretty weak interactions. So it's nothing to compare to classical antibody interactions. So very weak. So a non-wash assay like we have is very beneficial because if you wash, of course, and you have weak bindings, you can't detect this anymore, or you may just lose the binding. So this is really exciting. And we are trying to replace a lot of in-house ELISA assays that people do or chromatography methods, and we've made a huge progress over the last only 6-9 months. It's really amazing.
Wow, that's really exciting to see at what speed you're able to develop these new technologies. Now, let's take this a step further, Sebastian. Where do you see this space going in the next 5 years with all the technological advancements and new tools we have at our disposal?
If we're looking at developability, for example, it's a very emerging field. And what is interesting there, it's also driven a lot by AI, ML approaches that big pharma, for example, try to implement and to really come up with better drugs, better antibodies, maybe also from de novo design. So these methods need good data to predict. And these, at least for developability, these data does not exist. Maybe there is some data out there, but it's heavily biased. It's only antibodies and not those like bispecific and multispecific formats. So there's a huge need to feed good data into those models. And I don't know, but obviously there's a lot of hype about AI and machine learning, etc. But we'll see what will come out of this in the next 5 years. I'm really curious to see what will be possible. I don't know a lot about it, but we'll try to help with our assays to really make those tools more powerful, and we'll see how that develops. And I don't know in bioprocess development how much artificial intelligence is involved there. I haven't heard a lot about that yet, but I don't know. Did you?
What I'm hearing also from different people is that there will be a lot more integration with and combination of these different technologies between AI, using it for models, for integration or whatever it might be, or just the data analysis and then making informed decisions. So there's much more connectivity between these different tools probably emerging in the next couple of years or so. Sebastian, for our listeners who are new to this field, do you have any resources you could recommend so we understand the technology and also the importance of the CQO screening better?
For learning about our technology, our website is quite a good resource, I would say. And then more broadly speaking, about CQAs, I think you have to turn to the well-known journals out there. For CQA screening, I'm always a bit surprised that there is relatively little out there. For example, if we talk about glycan screening, there's not so much in terms of peer-reviewed publications. Maybe we should do more to this end, but yeah.
Maybe we should publish. We should create a publication. That's maybe the learning out of this. As we're wrapping up, Sebastian, what is the most important takeaway from our conversation?
I would think that it's really important that if you want to optimize a process, and this can be discovery or development, it's important to gather data which are robust and reliable on a lot of samples, not only the good ones, also the bad ones. And then from there really derive a conclusion which is really meaningful and that helps you to achieve a goal, which is ultimately to get a better antibody or better cell culture process, like we discussed, in combination with some modeling tools, whatever that may be, can really provide a good or a better outcome than what you would do just guessing what may be the right approach or the right conditions to, for example, run a cell culture. I guess that's really the way forward.
Fantastic. Thank you for highlighting this. Thank you so much for sharing your insights, Sebastian. Where can people connect with you?
I think the easiest way is to do it via LinkedIn. Of course, we're pretty active there, so that's a good way to get in touch.
Excellent. I'm going to leave the information in the show notes, so please do reach out to Sebastian. And thank you once again, Sebastian, for being on the show today.
Yeah, thanks, David. It was a pleasure talking to you.
Hey, smart biotech scientists. Thank you for tuning in today. What did you think about today's episode? Please tell us by leaving a review. I'm looking forward to hearing from you. And perhaps you're in the middle of a developability assessment or a screening exercise in process development. But you want to leverage your data set, you want to leverage your technology, but you're not so sure how to do that. Well, I want to offer you a free 30-minute assessment to help you get started and to help you make the most out of your experience. Just use the link down in the show notes and I see you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.